Proposed the mechanism of natural selection to explain the observable patterns of evolution. His views on these factors were heavily influenced by what we consider artificial selection, controlled breeding, hybridization. We don't tend to think of this kind of thing. We think technology, but it is. Technology is just the application of scientific knowledge to practical purposes. So we have bred organisms for traits that we prefer. Okay, for example, teosinte, which is the green plant on the top left, we bred into the modern corn. Or you take something like the wild mustard plant, and that's where broccoli, Brussels sprouts, kale, cauliflower, cabbage, all of these things came from. We bred them and controlled them for their various traits. Now, obviously, we've gone beyond artificial selection and selective breeding. We've moved into the realm of genetic engineering and direct manipulation of genes, which is still biotechnology, but obviously a much more complicated form. In some cases, whole animals are used to produce proteins instead of cells, and in some cases, we manipulate plants. But it doesn't really matter what you're trying to do. The plants on the far right, for example, have been genetically engineered to resist destructive insects, allowing for an increase in survival of these plants and increased nutrition for the population. The goat in the middle has been engineered to excrete human blood protein, antithrombin, in its milk. So we can use the milk to develop and make medicines for people that have possibly allergic reactions or maybe ineffective access to other things. So genetic modification is part of biotech, but so is artificial selection. And that's really where biotechnology started. So we've really been doing biotech for 100,000 years. When you move further in, the first thing you really have to do, though, is to extract DNA and RNA. DNA and RNA are nucleic acids. RNA is going to be a little more likely to break down. They're very unstable. And RNases, enzymes that digest RNA, are very common in nature and very difficult to turn off. So it's going to require the use of a variety of buffers and enzymes to inactivate those processes to preserve RNA. DNA is significantly easier to extract, and you can do it in a really rough way just by using detergent and alcohol, essentially. So you lyse the cell with detergent, you grind it up necessarily to sort of break it up, and then you separate it using ethanol, which it precipitates in. But you can also use more advanced forms that would use things like proteinase K, which is the picture on the right, which will digest any proteins that are part of that process, breaking down the histones and making the DNA easier to extract. Once you've actually extracted that DNA, you can move on to a variety of biotechnology mechanisms like DNA sequencing or polymerase chain reaction. There are a wide variety of DNA sequencing technologies that exist. It really kind of started with the Human Genome Project that used a three-stage approach to mapping the human genome. What they wanted to do was to develop linkage maps and then physical maps and then DNA sequencing. But we eventually realized that that was going to take a great deal of time, and other techniques have evolved. One of them is the so-called shotgun technique, where you cut the DNA into lots of little pieces. Those little pieces are easy to code together, and then we line them up using computer software. That has rapidly accelerated our technologies, but the problem is that there is a lot of repetitive DNA, as we talked about in a previous section. So knowledge of these sequences is very important, and other techniques are used to fill in those uncertain points. A slower but more accurate form would be the so-called chain termination method. The chain termination method uses a mixture of DNA nucleotides, that's deoxyribonucleic, as well as dideoxyribonucleic, so the DD NTPs there. And you mix and mix and mix, and it makes every possible alignment of the DNA, each one base longer. And then you take a laser and you find out the fluorescence that's triggered, and you can use that to code. So you make thousands and thousands and thousands of copies, you line them up so that each one is one base longer, and there you go. DNA sequencing can be done. Obviously, it's very, very slow, but it is much more accurate. More modern techniques have evolved in so-called next-generation DNA sequencing technologies. These developments are usually sequencing by synthesis. They've increased the spread and decreased the costs, but the new techniques, those fragments are sequenced at the same time to develop those processes. And then from that, we can then do things like proteomics, where we study the protein structure, and bioinformatics, where we look at the interrelationships, gene annotations, where we look for sequences that are gene-like, that can give us clues that can help us figure out the relationship. But these things are much faster, allowing the sequencing to be significantly more complex and significantly more efficient. Another technique that's really come into its own is utilizing that reverse transcriptase that we've talked about before. When you have an mRNA, it has already had the introns removed. So if you take the mRNA and you code the mRNA into DNA and then you sequence the DNA, you'll have the actual sequence of the gene without the introns. So any of those unnecessary components for making that very particular process has been removed. And this is what we did with like the COVID vaccine, for example. We took the mRNA for the spike protein. The spike protein is not infectious on its own, but it is a surface protein that can be recognized. And from that mRNA, we produced cDNA. And that cDNA is what was in the vaccine so that your body could learn how to respond without needing the viruses. So because it's mRNA derived, it's significantly more effective, but it requires this reverse transcriptase enzyme to make it possible. Where did that come from? We talked about this before. We do use it in retrotransposons, but it most likely evolved in retroviruses like HIV, for example, and then became integrated into our genome. Restriction enzymes are another tool in our toolbox here. Restriction enzymes are enzymes that have evolved to cut DNA. They cut them at very specific sequences called the restriction sites, and those sequences tend to be what's called palindromic. So if you look at this structure here, for example, okay, it's palindromic to the complement. So GAATTC, GAATTC, okay, that is going to be what I mean by the palindrome. The restriction enzyme will cut, it might cut it in a staggered formation, which is called a sticky end, or it might just cut straight down, which is called a blunt end, but that will open up the DNA. That open DNA can then be used to create what's called a recombinant organism. So if you take the plasmid, which is a small loop of extra chromosome DNA that's easy to exchange, and you cut it once with the restriction enzyme, and you take a gene that you want to insert and you cut it twice with the same restriction enzyme, those bases will match, and then all we need to do is add DNA ligase to put those strands together, and we've now made a recombinant organism that has both the original plasmid as well as the new DNA. That is something that's incredibly useful for things like gene cloning and medicine development and DNA libraries. It is one of the more essential techniques. Now, bacteria evolve these restriction enzymes as sort of an immunodefense. They do have sort of enzymes and structures that can help prevent them, like epigenetics, for example. If you have an epigenetic methyl group on the restriction site, then it can't cut. So there are ways to stop this process, but it's something that we take advantage of for biotechnology purposes. When you think biotech, you probably think gel electrophoresis. And gel electrophoresis is when DNA fragments are separated by size. DNA is negatively charged, so it moves towards the positive end of an electrical field, and so we run that through an agarose gel. And so basically the DNA is cut by restriction enzymes, and the pieces are separated from one another in a gel by running an electrical current through it. The negative charge, the segments are pulled to the positive end. Small pieces move through the gel faster than large pieces, and we typically use a dye or some sort of fluorescent marker to find those gels. Clumps of DNA build up, and we call those bands. You can also use electrophoresis to separate things like RNA or proteins. It's all based on the rate of movement. Just be sure you know what the charge is. If you're starting with a protein, for example, you might want to put the wells in the middle because some proteins are positive and some proteins are negative. Now, this will also depend on having a large amount of DNA because individual pieces of DNA are invisible. So you typically have to use large amounts or maybe clone the DNA or use polymerase chain reaction in order to have enough DNA to make it visible. Now, once you have the actual bands, you can then compare them. The old school process used to be so-called blotting techniques, where you literally blotted it to paper. It would look something like the middle picture here. So those gels were blotted to paper for saving and analysis, because the gels are made of agar. They're going to mold and they're going to break down. So the blot was able to be kept. And there's a whole lot of blots that exist. It started out with the so-called Southern blot technique. His name was Southern, so he called it Southern. And the naming convention kind of went from there. So you'll notice that they're all directional. It's a blotting compass. You don't need to know them. But the blotting techniques are used to identify the unique proteins and sequences that can then be analyzed through further processes. While things like polymerase chain reactions and microarrays and sequencing have largely replaced blots, it can still be cheaper and it can find positions of large fragments. So they can be useful, especially when you're looking at an unknown organism and you don't know what the sequence is, so you can't clone it because you don't know what to look for. So Southern blots haven't been regulated to trash yet, basically. They're still a very useful tool when you're beginning to explore a new organism or just when cost is a factor. From those banding patterns, you end up with different combinations. Those combinations can be used for things like forensics or paternity, in a process called DNA fingerprinting. Basically, a genome is the complete genetic material contained within an individual organism. And except for identical twins, every person's genome is unique. And because the probability of two individuals having the same DNA fingerprint is incredibly small, it provides compelling forensic evidence. The first case it was successfully used in was a rape case with Tommy Lee Andrews in 1987. Another case we can see here, for example, Earl Washington. He was accused and arrested. He had served 17 years in prison, and DNA evidence was pulled at a later point, and it turned out that it wasn't him. It was somebody else. And so what did they actually do? Well, restriction fragments are cut by restriction enzymes, and different people are going to have different lengths of restriction fragments. So we call those restriction fragment length polymorphisms. Why are they different from person to person? Well, the number of STRs, or short tandem repeats, which are non-transposable small segments of repetitive DNA, vary from person to person to person. And those are something that you inherit, and no two individuals are likely to have exact matches for all of those STRs. So we use polymerase chain reaction to amplify particular STRs and then run them through the gels. The probability that two people who are not identical twins have the same sets is vanishingly small, and that is something that we take advantage of. Now, you can't just do one or two STRs. You do typically a series of STRs, 10, 15, 20 that you check at once, as we've realized more and more that there are quite a lot of them. But because they are non-transposable, we know where to look for them, we know the sequence to look for, and it's something that we can code for. Now, I've mentioned polymerase chain reaction a couple of times because it's really important. Polymerase chain reaction is a method of essentially cloning genes without an organism being necessary. I could copy a gene in a bacteria, but I can't necessarily do that all the time. So polymerase chain reaction, the key ingredients are a so-called Taq polymerase, template DNA, primers, and nucleotides. So you're going to have a template DNA, that's going to be the base sequence. And you are going to denature that DNA by heating the DNA, separating the strands, because the heat is going to break the hydrogen bonds, which is going to provide single-stranded templates for the next step. Okay, you're going to add primers for what you want to clone. So you need to know the first couple of bases so that you can attach an artificially created primer. Then you're going to add Taq polymerase. Taq comes from a hot spring prokaryote, so it's a Thermus aquaticus. It's a hot spring thing, so it's able to function at these high temperatures when human polymerases wouldn't. Okay, that will then lead to tons and tons and tons of copies that you could use for a variety of techniques, like inserting into a plasmid, making a recombinant organism, or coding or electrophoresis, whatever you're going to be trying to do. PCR is very, very fast. Only minute amounts of DNA are needed to be present, and you can produce, after about 30 cycles, over 1 billion copies. Will all of them be copied correctly? No, there's going to be mistakes, there's going to be errors, mutations can occur, so there are occasional errors, but you can get lots of DNA from a very, very small sample. Recombinant screening can also be incredibly useful. Once you have made a plasmid that is recombinant, you can put that into an organism, and that organism will grow and develop. And as it copies its own DNA, the plasmid will copy. So this is another way of cloning, but it requires a bacteria. This process of inserting genes into an organism is called transformation, for the same idea, where you uptake genetic information from the environment. We're doing it by force, but it's the same kind of structure. So how do you know if you were successful? DNA is not visible to the eye, where you're going to typically use some sort of reporter gene. That reporter gene might be like an antibiotic resistance gene. So like, oh, I'm going to put in the gene for resistance to ampicillin, and then I'm going to grow the bacteria up on ampicillin. The bacteria that has this recombinant will grow. I'll know those were successful. More complicated and more advanced techniques will utilize fluorescent markers, like the example on the right. So now with fluorescent markers, you can look for multiple things at once, which is sort of more of an advanced operation. So basically, we label nucleotide acid probes that will hybridize and provide information, and that will show up as fluorescence, which is kind of cool. Another useful tool that's becoming much more common is a DNA microarray. A DNA microarray is a collection of microscopic DNA spots attached to a solid surface. Scientists use these microarrays to measure the expression levels of large numbers of genes simultaneously, or to genotype multiple regions of a genome. Each spot has a very small amount of DNA. Those are called probes. These can be short sections of a gene or some sort of component. And hybridization is detected and quantified by detection of fluorescence, typically, which we see on the left. The core principle between these processes is that basically they're complementary, they're going to match. Overall, this allows you to test lots of things incredibly quickly. In one example of a global expression study, researchers performed DNA microarray analysis of more than 90% of the genes of a nematode during every stage of its life cycle on a single chip. In addition to uncovering gene interactions, DNA microarrays can contribute to a better understanding of things like disease, new diagnostic techniques. Say, for example, if you could just put a drop of blood on a DNA microarray and test for, I don't know, all STDs or something, that is theoretically possible. So it's something that we're trying to figure out how to do. And then the last real form of biotech that we're talking about, which is far from the last real type, it's just the most basic, is organismal cloning. We've talked about cloning of genes without an organism, and we've talked about cloning of genes with an organism. But what if you're trying to clone an entire organism? So organismal cloning refers to this procedure of creating a new multicellular organism genetically identical to another one. It's essentially asexual reproduction. The term clone is used in a variety of mechanisms, but the most common form of cloning is what's called SCNT, or somatic cell nuclear transfer. You're going to take a somatic cell that has been differentiated, and you're going to put those genes into an enucleated egg, an empty egg. You apply a bit of UV light electricity to begin that process, depending on which you're doing, and it begins to develop. We know now that the less differentiated the cell is, the more likely cloning is to be successful. One of the more common examples is Dolly the sheep. You can see her on the far right. Dolly, which was a Finn Dorset ewe, was the first mammal to have been successfully cloned from an adult somatic cell. Dolly was formed by taking a cell from the udder of her 6-year-old biological mother, and Dolly's embryo was created by taking the cell and inserting it into a sheep ovum. Now, we're really bad at it. It took 434 attempts before Dolly was successful. The embryo was then placed inside a female sheep that went through a normal pregnancy. And the cat in the middle seems kind of random, but this is the first cloned cat. It was named CC for carbon copy, and they did the same sort of process. But you'll notice that the fur patterns are not the same. Why? Because the fur patterns of these tortoiseshell cats comes from random exon activation. So even though you can be a clone, you may not be identical. Maybe nurture, maybe nature, maybe random mutations, all sorts of issues. Now, plants are significantly easier to clone. Whole plants have been cloned from a single differentiated cell since the 50s. We've been doing it for all sorts of structures. Students have found and researchers have found that since plants maintain their totipotency through their entire lifespan, that sort of process is significantly more effective. So we can take a cell from anywhere in a plant and we can clone the plant. Whole industries have developed around that process, and most modern like plant nurseries and farms will have plant cloning labs. Doesn't take much to do. It's actually really simple, and it can be very effective. So this is sort of a brief overview of our biotechnology toolbox, and I hope it helps.